moment. Okay, now already. Okay, good evening, everyone, and thanks for your attendance. And today's Asian Pharmaceutical Online Symposium. And so welcome Professor He and Dr. Zhao Liang and also Professor Charley Wu. And so, and before we start our symposium, and so first I want to share two videos with us. The first one, it is the video is about the AJPS, and it is our journals. Okay, and this is first video. This is our AJPS journals and Asian Journal of Pharmaceutical Science. And the second video is about our Computational Pharmacy Society. And just wait a moment.
The Earth, a prosperous planet, is home to trillions of creatures. Diversity lives in the ocean, land, and sky, makes the world lively and vibrant. However, life suffers from disease and death, like a joke made by nature. But we human beings fight against destiny. Since ancient times, we've used herbal medicine to fight against diseases. In the last 200 years, with the rapid development of science and technology, we learned about cells and organisms, as well as molecules and atoms. And scientists hunt new drugs from experiments. Nowadays, the development of computers and the internet have drastically changed the way we live, work, and even think. In drug discovery and development, we apply artificial intelligence to analyze the potential of drug delivery, to screen formulations, and find the best solutions. We can also perform molecular modeling to stimulate the molecular interaction between small molecule drugs or protein drugs, or even gene design drugs and the carriers. We can further build a virtual human body to simulate the behavior and therapeutic effect of drugs. Computational pharmacy shifts the paradigm from traditional trial and error experiments to computer modeling and data-driven AI, and speeds up drug discovery and development, and then benefits the academia, pharmaceutical industry, and patients. The future is already here. It's just not very evenly distributed.
Okay, and so Okay, and now we welcome Professor He and the editor-in-chief of AJPS, Asian J Pharmaceutic Science, to give us the opening speech. So welcome, Professor He.
好，谢谢欧阳，谢谢欧阳。我就今天我们主要还是中国人，我的英文口语也不是特别好，我就用英文讲了。那我们今年的这个因为特殊疫情关系，本来我们应该是在今年我们应该在韩国有一次亚洲药剂学论坛，因为疫情的原因，国际会议呢都停止了，没有办法开，所以呢我们也想到一个但是我们这种交流，我们这种学术交流是不会停止，所以呢我们今天呢是我们整个学术交流第一场云交流网上交流活动呢，今天的主要活动呢由欧阳教授呢来主持，那么作为我们今天的主持嘉宾，我们也今天有3位教授来做我们的今天的这个有赵亮教授，还有邬传宇教授那么赵博士呢是 Ph.D 专家，那邬教授呢都是计算药剂学方面的专家。后面的话呢，有欧阳教授呢会给大家一个详细介绍。那我们今天的计算药剂学呢，实际上呢，在药剂学药品开发研究过程中间呢，起到非常重要的作用。大家对这个方面的这个领域兴趣也越来越多。今天的这个主题呢，主要就是计算药剂学。欧阳是这个方面的话研究非常活跃的一个专家，也非常高兴的介绍呢，欧阳也是我们省药的校友，也是我们 AJPS 杂志的编委。那么也欢迎，非常感谢呢大家欧阳教授和邬教授还有赵博士对我们这个杂志的支持，对亚洲药剂学论坛的支持。那么也欢迎各位专家各位老师，也欢迎各位专家各位老师呢以后继续呢积极的参与包括主持我们这种亚洲药剂学的这种分论坛，云上的这种网上的这种分论坛那么我们以后的话可能每一次呢有一个不同的主题不定期的经常性的组织这样一些活动。我们期望呢这个疫情呢能够尽快的结束，那么大家的生活能够恢复常态，这样我们下一届的亚洲药剂学论坛的正式的线下的会议我们就能够召开，也希望大家能够有机会呢在不远的将来我们就能够进一步的在线下进行交流。但是线上的这个交流呢我们会间断，经常会举行那么再次感谢欧阳教授的这个辛苦的主持今天的这个活动下面的这个会议呢就请欧阳来主持谢谢欧阳
好，非常感谢何教授的支持。然后我先说明一下，因为今天我们这个会议因为是面向全世界的海外的，所以我们的3个报告都是用英文来讲。后面的每个报告是半个小时30分钟，然后后面是留将近1个小时的讨论的时间，会全用中文的，然后大家也可以用中文的后面提问在后台留言，好吧，谢谢。Okay, now we first introduce our first speaker. It is Dr. Liang Zhao. And Dr. Liang Zhao is the current director of the Division of Quantitative Method. Okay, and Dr. Liang Zhao is currently the director of the Division of Quantitative Method and Modeling at FDA. Dr. Zhao have wide scientific and management experience from industry and regulatory agency. Through his 16 years professional career, he has developed his leadership in industry R&D, quantitative method and modeling, and model-based strategy decision-making in regulatory and industry setting for generic and new drugs. And Dr. Zhao have already published over 70 peer-reviewed articles, 5 book chapters, and over 100 conference abstracts, and delivered over 60 talks in different workshops, international conference. And Dr. Zhao received his Ph.D. in Pharmaceutical Science and a Master in Ohio State University and a Master in Pharmaceuticals from Shanghai Medical University, and also bachelor degree in Pharmaceuticals from China Pharmaceutical Universities, and also MBA from University of Cambridge. So welcome, Professor. Welcome, Dr. Zhao.
Thank you, Dr. Ouyang, and thank you, Professor He, for the introduction and the invite. As mentioned earlier by Dr. Ouyang, so my presentation will be in English. However, in the Q&A session, we'll convert to Chinese. 大家好，大家晚上好，我这边是早上7点钟，我们先开始用，因为是一个international event， 所以先用英文来讲我的报告。如果有听不懂的什么地方，我们在那个 Q&A session， 到时候我们用中文交流，希望大家包涵。Ouyang, can you see my slides?
Yes, I can. Yeah.
Okay. So as Professor Ouyang mentioned, I'm currently the director for Division Quantitative Method and Modeling, Office Research Standard, Office of Generic Drugs. Some of our duties just to, every year we have about 25 million user fee related research fund that being allocated to the office to support GDUFA sponsored research. GDUFA is more like the user fee program for generic drugs. And the research outcomes can certainly benefit both new drug and generic drug development. On top of that, we are also been meeting with the industry to discuss complex issues. Once we have a complex issue in development have been identified, then we'll establish a project, also allocate some funding opportunities throughout the globe. So the best lab, the best scientists can collaborate with us and rule out the best result. So my presentation has been cleared by FDA. And however, the presentation represents the views and the perspectives of the speaker, myself, and does not necessarily reflect the views of US FDA. So the agenda today, I will do a quick introduction. Following my introduction, I'll do, based on the focus of the theme of today's meeting, I'm going to share with everyone in the audience about challenges and opportunities in regulatory science for computational pharmaceutics. From that, I want to mainly to share a financial year 2020 generic drug regulatory science initiative public workshop on May 4th, 2020. So we have invited a panel of top-notch scientists, professors, drug developers in the panel to discuss if we want to develop advanced computational pharmaceutics and also develop a lean program, make a drug quickly to the market, what will be the scientific barriers? So after download some expert opinion in that, so I will do a summary. So what's computational pharmaceutics? This is what I got from Wikipedia, and also checked Dr. Ouyang's publications. Seems he's the leading expert in this field, and also he come up with the terminology, which I'm very impressed. Computational is about computational characterization of drug delivery and formulation development. The regulatory impact include bioequivalence establishment for new formulations, local drug delivery, and manufacturing control. It's very conventional area, but now if we instill modeling and simulation, then we are facing a new branch of science. Key questions that we have viewed in the FDA May 4th public workshop include 3 bullet points. The first one, how to evaluate the data from in vitro studies and which in vivo studies are clinically relevant. With that, then we can justify if there's a Q1, Q2, Q3 division for equivalence assessment. Can we use modeling simulation to tell FDA or tell the Chinese agency or European agency, okay, it doesn't matter. So because we are very confident based on this model, then we'll evaluate the modeling result and say, okay, your formulation is fit for the purpose, then we can accept your application. The second question, what are the challenges for industry in implementing modeling simulation method to support more efficient regulatory BE pathways? So we have a good theory, but how can we make it work from industry? So at FDA, we kind of are the regulatory reviewers. We do not have the first-hand experience. Then we would like to hear from industry people, from academia, what are the part that's blocking them have a quick rollout of applications. The third question is, what are the emerging expertise tools in implementing new BE approaches? So emerging expertise, I've also did some literature research for computational pharmaceutics. I see Professor Ouyang focus on big data, machine learning. That's certainly one of the emerging expertise. We are also sponsoring some research in FDA on top of that. Actually, my group have published the first 2 papers in the field of pharmacometrics using machine learning to deal with survival data to predict when a new drug is approved, when the generic will be in-house at FDA. In case we have been talking about a Q1, Q2, Q3, in case you don't know what's Q1, Q2, Q3, here is the labeling for them. Q1 is qualitative sameness. Q2 represent quantitative sameness. Q3 is just the macro structure, physical chemical sameness, similarity that certainly include in vitro dissolution, which you may be familiar with. So in the expert panel, you can appreciate like a good assembly of the world-renowned expert, and that range from universities. I think you may recognize Dr. Amin Rastami. He's more like the godfather of SimSip. Now he's a kind of a president at Certara. Andrew Hooker, and he's one of the top pharmacometrician in the world. He's closely collaborating with Dr. Mats Carlson in Uppsala University, Sweden. So Charley and Glennis, Sandra, Vera, they are all representing the industry scientists who are developing the formulations and developing generic drugs, and also help with the consulting for new drug development. Stella is the division director from Office of Biostatistics. Stephen from University of Florida. Sid Bupani is more like a formulation scientist from absorption systems. Raj from Sandoz. Certainly Lucy Fang is a associate director in the division, in my division. So every year we will hold a workshop to solicit ideas, to get ideas from the industry, from academia. What you think there's a scientific gap? In the May 4th meeting, we're focusing on research ideas for financial year 2021. So I want to give a quick review for the research focus we have conducted in financial year 2020, which covers from 2019, the second half of 2019 to the first half of 2020. So it includes complex active ingredients, formulations, or dosage forms. For complex active ingredients, certainly you can think about the Chinese herbal medicine, where is the API, the active pharmaceutical ingredient, is not a single entity, it's a mixture of things. Actually, there are many peptide products the mixture of things. So if you want to say the peptide product, they are the between the reference and the generic is the same, how can you evaluate the sameness? We need to develop some quantitative approaches. So we have published something like using high dimensional reduction technique, like a principal component analysis to make it feasible to evaluate the sameness of a complex API. So we have been focused on complex route of drug delivery, complex drug device combinations, and tools and methodologies for bioequivalence and substitutability evaluation that include trial simulation, BE study simulation, PBPK. PBPK represents physiologically based PK modeling, and PK/PD modeling, which is the conventional area of pharmacometrics where I'm from. So we are also focusing on the expansion of BCS class 3 biowaiver. Currently, if you have a BCS1 compound, basically the regulatory pathway to get another, say, generic or new drug on the market under 505(b)(2 is very easy because it's a BCS1. You don't need to conduct many PK studies. Even you don't need to conduct any PK studies after you can prove the product. However, if a BCS3, then if you want to have the same treatment, you don't want to conduct any studies in human, then you certainly need more in vitro assessment and justifies the BCS3 drug. And the modeling will have a very critical impact on that. The fourth thing is develop method and integrated technical solutions that allow FDA to leverage large data set. This is the machine learning part I'm talking about. Currently, everyone is talking about real world evidence. Can we use real world evidence to approve new drug? Certainly for generic drugs. So when two products are on the market, are they performing the same? Then we can collect patient data that can include electronic health record, health insurance claim data. We have a huge data set. Then from the database, we evaluate the performance of the product in the post-marketing stage. So I mentioned earlier that 3 bullet point questions we wanted to address. The first question is how to evaluate data from in vitro studies and which in vitro studies are clinically relevant. So we are talking about, I think in my school time in China, I think the concept about a formulation is about mainly focusing on quality, QC, control. However, there's another element, whether the formulation property or attribute, whether they are clinically relevant, whether if you give that to human, will they affect the PK, the drug exposure on the side of action, and the clinical performance, whether they can affect the drug effect. So if we can figure out that relationship, then we can basically have some influence on the safe space, what will be the spec for Q1, Q2, and Q3. Here are the current challenges. For in vitro only approaches, narrow Q1, Q2, Q3 standards often requires let me change my pointer a little bit. Okay, so often requires exhaustive reverse engineer of the reference product using state-of-art analytical techniques. So even though the reverse engineering is a highly developed technique, however, believe it or not, even in the US, in the Europe, the generic industry cannot figure out some complex formulations for the brand name drug. And that way they can really not make the product Q1, Q2, and Q3. Sometimes the patent can block the ability to use a Q1, Q2, Q3 formulation. If the brand name product are smart enough, they just patent everything. That make others difficult to copy. They can sue you if you copy, and the other industry will face big financial risk if they have a failure in the court. A significant need to develop modeling approaches to justify relaxation. Relaxation meaning can we expand the spec, so make it easier for regarding the regulation of compositional refers to Q1, Q2, and macro structural requirement. Macro structural referring to Q3 requirement. For example, can we use IVC as supported by PBPK model? The conventional IVC just you have in vitro parameter change, then you have a human being PK difference observation, then you empirically establish the relationship. Now we want to mechanistically establish the relationship using PBPK model. So once you have established the relationship, basically you arbitrarily change Q1 parameter, Q2 parameter, Q3 parameter, then you can expect the change in human body and the drug release in human body, the drug PK profile in human body. Then there you can say, okay, even though I don't meet the FDA standard, the deviation, the difference is by this much, but it doesn't matter because it doesn't affect the human PK exposure. You know, certainly people, once they have the relationship established, you can build that argument. Then we also want to setting up excipient exception categories for class 3 biowaivers. For class 3 biowaiver, excipient 辅料 is also something we need to put into consideration, whether excipient can affect the drug absorption and by how much, what excipient are safe, which are not, which excipient can affect the transporter or metabolism of a drug. Those are very, very important. Okay. So then we just published the thought from the breakout session, and it's a clinical pharmacology therapeutic paper, and we summarized the thinking from the question is that if we want to make a product, what do we consider is the API properties, the manufacturing process, and the formulation composition. This is a product. Then from this product, we can evaluate the Q3 properties of the product. That include the biopredictive in vitro release, dissolution testing. With that, then based on the relation, the PBPK understanding and the PKPD understanding and the ER relationship, exposure response relationship for clinical performance of the drug, we can bridge the Q1, Q2, Q3 properties with the drug performance. Therefore, we can infer what is the safe space for Q1, Q2, Q3 properties. You know, that's the thought I just illustrated. Regarding with BCS class 3 biowaiver, this represent ICH-M9 guidance. I believe, you know, I do not have good assessments of the audience profile, the knowledge base. However, say if you want to waive in vivo human being study, this is the flow chart you must follow. Say, are there excipients in the formulation with known or suspected effects on drug absorption? If no, then are they Q1, Q2, Q3 similar or the same? If yes, then you don't need do any study. You just make a formulation and the drug will be on the market. However, if you violate this kind of conditions, you will see if the excipient difference is within 10%. If no, then we are not going to grant bio waiver. However, if you have a good modeling understanding of the relationship, you can make present your case to us, to FDA, then we can evaluate that and you can make a justification. So the gain is that if you have a model, you don't need to do a human study if the model has been properly developed. Here is another slide talking about some details of the BCF3 oral drug product bio waiver. These are the details to qualify a bio waiver without a modeling. So that's for orally administered drug. For locally acting product, we consider them as complex formulation, complex generics, because you cannot evaluate bioequivalence, or even for new drug development, if you have a detection of drug concentration exposure in human body, and that doesn't represent the drug exposure at the site of action. For example, if the drug for the eye is a complex ophthalmic drug, then first of all, many times you cannot detect any drug concentration in human blood. Secondly, even you have a detection of a drug concentration in human blood, serum, plasma, they does not represent what the eye drug exposure. So whether it will be effective, whether it will be delivered to the site of action. So with that, we have a Q1, Q2, Q3, and the release, in vitro release. Then we need to build another layer of relevant tools, which include the PBPK. Then we link all the Q1, Q2, Q3 parameters to the local drug delivery at the site of action. If this has been understood to a certain extent and the FDA is satisfactory with the result, you do not need to conduct a clinical study. Certainly that can supported by other clinical study results. So not necessarily the primary clinical endpoint. Say it could be a PD endpoint, which is sensitive to drug delivery change. Could have, in this case, could be intraocular pressure measurement. 眼压也可以代表这个药物的一个疗效。So you can use those things to evaluate the bioequivalence as well. For this question, the panel think the potential challenges for ophthalmic product is that an alternative in vitro model informed B approach for chronic complex ophthalmic products. So currently, in the ideal world, we just do a study in vitro, then we invest a little bit in vitro and not invest too much on human being, and with the modeling, we can prove product. However, there's agreement that establishing RV-RVC for the identified ophthalmic product, CQA represents critical quality attribute, is difficult. In many times is because you may have abundant in vitro data. However, you don't have human, the in vivo data in human body. That way, you really cannot establish a RV-RVC relationship. Sometimes the cost to establish a RV-RVC relationship is higher than the cost you can just go directly conducting a human study. Here, the modeling or computational pharmacometrics or pharmaceutics can really bridge the gap. So the challenges from industry perspective overall. So we talked about the drug orally administered, we talked about drug locally acting. So overall, from the panel, people think streamlined B approach have in principle been welcomed by the industry. Streamlined approach refers to that you use in vitro only approach, and then bring a drug on the market. In theory, sounds good, but in practice, very difficult. Meeting compositional Q1, Q2, and Q3 requirements can be challenging in many cases. The challenges exist in in BCS3 drugs, and also challenges in conducting comparative clinical endpoint B studies for locally acting drug product. So here's another concept you may want to know. So if the drug is locally acting, so the PK exposure in human body may not reflect the local drug concentration, cannot be used for drug approval. Therefore, but if the drug is orally inhaled, so the site of action is the lung. If it's an ophthalmic drug, the site of action is the human eyes. So if the dermal product, the action site of is the skin, but it's really difficult to get PK concentration measured in your eye, in your lung, in your skin. In that regard, the last resort is to conduct clinical endpoint B studies, because we don't have any means to evaluate the drug delivery anymore. Then we just, okay, let's see what the clinical response. If they are equivalent, then we'll approve the drug. But the clinical endpoint B study is not sensitive. You may have heard stories from biosimilar, 生仿药. Even you double the dose of a drug, the clinical response stays the same. 就是你把剂量就是 double 双倍之后，也不能在临床上检测出有任何差异。Therefore, the clinical endpoint B studies is not that sensitive, cannot be reliably or sensitively used for to detect the local drug delivery. Sponsors sometimes shy away from developing their own novel streamlined B pathways for similar reasons. Dilemma between extreme B approaches with too much or too little sensitivity. If you are using Q1, Q2, Q3 approaches, we are requesting a very sensitive, a too stringent approach, even though it's in vitro only. But if we are requesting a clinical endpoint study, it's too insensitive. It's not sensitive to the dose change. Let me check my time. Okay, my time is always up, so I will speed up just to illustrate the main point in the following slides. However, there are opportunities from the industry perspective has been downloaded in the workshop that include significant opportunity exists to apply modeling method to justify relaxation of the requirement. Many reference product drugs are locally acting drugs and have to have too few or no approved generic drugs, at least in part because of the difficulties in qualifying for streamlined BE pathways. Opportunity to facilitate the development of generics by relaxing the compositional macrostructural criteria to qualify streamlined B approaches using modeling method to probe the effect of divisions from narrow Q1, Q2 criteria and impacted the product performance in vivo. So they all propose we need to put more money in the research in this area. Let FDA spend more money to sponsor outside the best labs to conduct more research. Certainly, we have been constantly doing that. So this is a quick summary. Given the time, I won't repeat again. So the second key question is, what are the challenges for industry in implementing modeling and simulation method to support more efficient regulatory B pathways? Here, we are talking about, you just heard about the PBPK model, right? For example, if PBPK model is used to support the drug development, what are the difficulties in implementing it. So there are many pharmaceutical companies where model-informed drug development is not a common practice, or it's not even known as a path for drug development. Here, if we talk about MIDD, which is a concept popularly used in new drug development. However, even in the new drug development, there are many companies, for smaller companies, they do not have the group of scientists who can really develop a model, including a PBPK model to support the drug development. I think in China, in most of Asian countries, the model application is still very limited. It's not a expertise everyone have. So first of all, there's a knowledge gap. Secondly, the virtual bee requirement for prudent use of PBPK in uncharted territories. Even we have the right expertise, we have the modeler who can conduct virtual bee simulations. Virtual bee simulation meaning if we understand everything, we integrate the knowledge and data. We just use computer to simulate a bee study or other study, a trial simulation. However, there are many gaps because we do not know many system parameters, say what the skin permeability in different layers, what the biodistribution of drug in your eye, what the API properties, how to understand the inter-occasion variability, meaning if you gave the drug to the same individual in different days, today I have a drug exposure of 10, tomorrow I have a drug exposure of 12. There's a inter-occasion variability even within the same individual. In that case, how can we use the model to characterize the uncertainty, the effect of the excipient on the physiological system, effect, what the 辅料的作用. So currently in the PBPK model, these are the mostly evaluated parameters that are being investigated by the experts and published in literatures, where people have evaluated whether the solubility of the API, the partition coefficient, particle size distribution, gastric emptying time, 胃的滞留时间，等等等等, whether this will affect the drug exposure. However, there is a new thought from that okay, for the sensitivity analysis, you cannot just change one parameter at each time. Many parameters are auto-correlated. For example, the liver blood flow is closely correlated with the liver volume. There are other known physiological correlations like the gastric emptying time is correlated with the gastric pH. The gastric emptying time is correlated with the particle size. So in PBPK model, there are some thinking that you have in vitro testing for locally acting products for topical transdermal products, or you need to understand the skin physiology and how the drug can penetrate through different skins. Then you have all the physiological parameters figured out. I think SimSip has done a good job. In also gastropolar, there are several vendors in the field. Their business is to figure out all these parameters. Then they can predict what will be the drug exposure once given to the human body. You can extrapolate from healthy volunteers. It could be Caucasian, the white people, to the Asian people, to the Black people, to Indian people, to the diseased population. Everyone has a different skin, right? And also to the elderly subject, when people, for people with older age, they may have different skin parameters. For the children, they also have different skin parameters. We need to figure all this out to make sure that once we approve a product, there's no other risks with the specific populations. And certainly we can engage the Bayesian population if we information if we want to develop PBPK model. However, we cannot blindly trust a PBPK model. The model needs to be calibrated and being tested with different formulations. Then with that, you can simulate a clinical study. So I will skip this slide given the time, but I want to also mention that in another area in the long-acting injectables, they are billion-dollar drugs, but there's no currently even no generic drug on the market because current regulation require they be conducted in the patient with the steady state. That means you need do a study like a year long or a 2 year long. So which is almost impossible for the industry to bring a long-acting injectable to the market. However, the model, once we can, this is from Uppsala contract we are conducting with them. Their idea is that if we have B data, we have models, the models could from the new drug side, when I develop a new drug, we already figure out the modeling structure, then we feed the model with the new data. Once the model has been sufficiently validated, verified, we can conduct the trial simulations. We can conduct 1,000 trial simulations. Then determine the distribution of the ratio mean and make a assessment. The same concept can be applied to the new drug development. And this is something ongoing, the worth noting. And this shows some data here. Type 1 error plot shows, okay, if you use the model-based approach as compared to the conventional approach, the model-based approach actually have a very good control of type 1 error. Type 1 error means the error to approve a product, which is not a bioequivalent. So we don't want to increase any risk of approve false product. And also compare the power to make a B pass based on the model for the model-based approach as compared to the conventional NCI-based approach. NCA, non-compartment analysis-based approach. Here, we are talking about if the drug is bioequivalence, if it is a good product. So if we use a model-based approach, then the chance to make it a B is, as you can appreciate from here in the red dot represent a model-based B approach, the chance to approve their BE is much higher than using the conventional NCA approach. This is good to the industry, right? If you are the industry, if you have a product and you want to approve that, now there are two approaches. If you use the model-based approach, if your product is good, then you have a higher chance to get your product to the market. You have a earlier, you can enjoy a profit margin earlier, right? That's the benefit to the industry. The type 1 error is benefit to the FDA. We don't want to prove bad product. Here, to the industry, you want to have a higher chance to let a good product to be approved. So with that, I talked about in vitro, in vivo, PBPK model, PK simulation. We only talked about the PK, right? Down the stream, we will have clinical response, which is a model type called exposure response analysis. If we have a drug concentration, what will be the clinical effect? So can we link the clinical effect all the way back to the formulation design? From here, the end to the beginning. So if we can establish the connection, then we can certainly, even from the beginning, when you are design a formulation, if you are developing a manufacturing process, then if you can figure out all the parameters that correlated with the clinical response, then you can have a better control and have a better success rate in drug development. So with that, I think my time is up. So I will skip the rest of the presentation. Here I want to mention here in the case that Bikram, where you can figure out, okay, the critical saturation ratio or the precipitation rate constant, which can be measured in vitro, can predict the drug exposure in human body. With that, I want to give my summary at the end. The computational approaches represent a key component in the current landscape of regulatory science need. And conventional and novel PBPK, PK/PD exposure response models will be critical tool set in guiding computational pharmaceutics. I want to acknowledge some colleagues in FDA, as Ouyang mentioned, introduced at the beginning. I do have thank the colleagues in the new drug side. I've worked some of the thought and development really gained from the whole career. You know, initially I worked with the pharmaceutical industry, then I joined FDA to the new drug side, served as team leader for pharmacometrics, then transferred to transition to generic drugs. So with that, I want to thank you, everyone. Thank you, the audience. I'm looking forward to talk with you again in Chinese in the panel discussion. Thanks.
Okay, and thanks a lot for your fantastic presentation, Dr. Zhao, and thanks a lot. Yeah. Yeah. Okay. And thanks, Professor Dr. Zhao. And now we are second speaker, and Professor Charley Wu. And now I will give a brief introduction to Professor Charley Wu. Professor Charley Wu is a professor in the Department of Chemical and Process Engineering at the University of Surrey, UK. And he is an editor for Powder Technology, is a leading journal on particle systems. He also published in 2 books and also over 100 scientific papers. He also give over 60 invited presentations. And Professor Wu also is a member of the editorial board, such as particularly Pharmaceutical Syndicate and the Journal of Engineering. And also Professor Wu, his research is widely supported by the global pharmaceutical companies such as Pfizer, AstraZeneca, Genentech, Sanofi, MSD, and also the European Funding Agency and also the British Funding Agency. So welcome, Charley. Yeah.
Right. Thank you, Professor Ouyang. Good evening, everyone. So I will share my screen. Ouyang, can you see my screen now?
Yeah, I can see. Yeah.
Is the projected screen or is the presenter view? I got is it normal one? Or?
I think it is projector one.
All right. Okay.
It is fine. Yeah.
Okay. So there's a 2 I can swap. Just oh, sorry. So how about now?
Yeah, I think it's fine. Yeah.
Yeah. Oh, okay, good. We'll make a start. Firstly, I would like to thank Professor Ouyang and Professor for inviting me. Also thank AJPS and the Computational Pharmaceutical Society for giving me this opportunity to talk about the research we have been working on using the computational tool in pharmaceutical product development. So firstly, I will give a brief explanation why we needed to develop computational tools for pharmaceutical product development. Then I will introduce a couple of computational tool or model we have been using in collaboration with a pharmaceutical company, trying to understand the manufacture process and understand the formulation development. So I'll give you some case study. Firstly, I know most of us know that the drug discovery process is quite long and expensive exercise. So you start from 10 to 20,000 candidate drug through a series of screening and optimization. If you are lucky enough, you will end up one drug that you can make into the market. Then hopefully we'll make a lot of profit. So during this process, the formulation development, also process development, product development plays quite a significant role, because you needed to evaluate the efficacy of your product. You have to make them into certain products. So once you identify this drug you want to manufacture it, then you're going to make sure the manufacture process has been designed properly, so you can make products efficiently with specified quality. So in this talk, I will mainly focus on solid dosage forms, because this occupies like 80% of the all medicines on the pharmaceutical market. So during this process, drug discovery process, there's 75% of the cost you put in actually is paying for the failure. So this has been used to identify those we can't make them into a market, those formulation or drug candidate that we can't make into the candidate. So here shows some statistics. So the cost increased dramatically. So now a typical new drug may cost about 1.2 billion pounds to development. Also it takes about 10 to 12 years. So it's quite a long process. So in order to reduce the R&D, research and development cost, pharmaceutical industry are exploring different strategies. So the typical strategy including the first is that more tightly managed clinical trial, where everyone knows that it costs a fortune to do a clinical trial and quite expensive process. Also, the second strategy is that to develop new and innovative approach to R&D. That is crucial for future pharmaceutical manufacturing. One additional strategy is that trying to make a decision far earlier in the process. So if you know drug candidate can't make in the market earlier, you will save a lot of manpower, save a lot of time and money in pursuing this particular drug candidate. So to achieve this, the computational approach can play a significant role in this process. So in this drug discovery practice, the challenge is not only just finding the best molecules that you can make a product onto the market and deliver to patient, but often a majority of the exercise, the practice is trying to filter out the better candidates. So if we can identify those substances that can't make into the product, if we can fill faster, fill earlier, then we can fill cheaply. So that's why people have been starting to talk about the 0 gram strategy will be most efficient if we can do some kind of screening on the exploration on computer before you actually running large experimental campaign in the laboratory, then it will save a lot of money. So, so the idea word is that we would like to have a kind of a tool that we can evaluate the performance, processability, and the product properties of identified drug molecule at the very early stage of the drug development, and just use only a very limited amount of material. You know, at the earliest stage of the drug development, normally you have a very small amount of material. If we can do some evaluation based on this limited amount of material, that would be really beneficial. Also based on smaller amount of experiment, so that we can obtain optimal formulations, also can design an effective manufacture process, so that we can actually reduce the duration and the cost in drug development. Obviously, this is a multi-scale problem. You go from molecular to API particles, and then eventually from API particle to kind of products. So in my talk, I will mainly focus on from API particles, how we can go from API particle and explore how those API can be actually made into kind of products. So it's a solid dosage form we're focused on in this talk. So if we look at the pharmaceutical tablet as an example, so we know most of the medicine actually in the tablet form. So for identify the API, so the drug, ideal situation is that we just mix the API with some excipients like filler, disintegrant, binder, and glidant, mix them. Also, we can add some lubricant to facilitate the compaction. So we put the formulation into a kind of a tableting system. And then we can make a tablets, and we can then coat it and pack those tablets, and we can put onto market. So that's the idea, the optimal manufacture process. However, for most of APIs, most of the drug particles, they're either too big or very small, or too small and very cohesive, that you can't really mix them very well with the excipient. You can't put them into a manufacture process. In that case, we have to use some kind of a granulation process. So for the granulation process, there's two types of approach. So one is dry granulation. The second one is wet granulation process. So in dry granulation, you mix your API with some excipients. You put some kind of a pressurized agglomeration process, typically like a raw compaction, and then produce ribbon or flake. You break the ribbon and flake into granules. Then you mix the granule with disintegrant lubricant and put in tablet machine. So this is particularly useful for APIs is sensitive to moisture and heat. We can also use wet granulation. So instead of using dry granulation, you can we can put a liquid binder into the process, and then this liquid binder will glue fine particle together. So then we dry the liquid out, and then we can use the milling to control the granule size. We mix the granules with the binders and the other excipients. Then eventually we can make tablets. Then in the typical manufacturing process, it will involve different system and different unit operations for mixing, for granulation, for drying, and so on. So typically for identify the API, the formulation development and the process design is not a trivial task. We needed to ensure each unit operation is performed properly. Conventionally, we can use this try and error until we got a proper formulation and manufacture process. That's very time consuming and costing. And to some extent, the pharmaceutical industry lag behind many other industries in exploring innovative tools like a computational approach and AI in product development. This is partly due to the challenge behavior of formulations. Because formulation sometimes it can behave like solid, it can behave like a liquid, or can behave like a gas, as the video just showing here. So we can't just using a single model to simulate the entire manufacture process, and we have to consider the diverse powder behavior in different unit operations. So there's no model, no single model that can fit all requirements. Then there's different computational tool has been explored in the literature, which including CFD, computational fluid dynamics, discrete element method, finite element method, also hybrid or coupled DEM, CFD, DEM, FEM. Also now increasingly artificial intelligence and machine learning has been used in formulation and product development as well. So I will introduce some of this model today. So the first one I would like to talk about, this one I have been working over the last 2 decades or so, is on the DEM, discrete element based method. We also couple DEM with CFD or with other approach as well, like a lattice Boltzmann method. So just very briefly, in DEM, we treat the formulation as the individual particles, and we look at the motion of individual particle in the process. So we here we'll talk about probably a model of millions or billions of particles in the process. We can also couple DEM with a fluid server like a CFD to look at the surrounding fluid or gas, how they affect the motion of the particles. So what we can do with DEM, so use DEM, we can look at mechanistic understanding. For example, here shows an example we using DEM CFD to look at how the API particle is going to stick on the so how API particle. So I click the wrong button to. Right. So here we show the example how we using DEM CFD to look at how API particle is going to stick on a carrier particle during mixing process for dry powder inhalation formulation development. Also, we can explore how the API particle initially stick on the carrier will be detached from the carrier and dispersed. So we can use DEM CFD for mechanistic understanding. We can also using DEM to look at the mechanical characterization. So for example, we know for pharmaceutical tablets, we typically using diametric compression to measure the strength of the tablets. By using DEM, we can run exactly same exercise, same test. We can look at diametric compression. We can also run 3-point bend test to measure the strength of tablets or compressed material. What we can do then is we can relate how the tensile strength of your tablets is related to the porosity. So how dense your material is going to compress, and how the effect of surface energy will affect. For example, if you said you want to make a tablet to reach a tensile strength of 2 megapascal, so then we can use this chart to identify at which porosity you need to compress your tablets to, and what's the surface tension you need to surface energy you need to have for your formulation in order to achieve a tablet with 2 megapascal. So by using DEM, we can establish this kind of material maps, and that will help formulation design. So that's another application of DM and CFD. Certainly, we can also use in DM to look at the process modeling. So this one shows an example we did recently on twin screw granulation. So we can actually model the actual process with the particles, and we will look at how particle of different property or different process condition, like the rotation speed of the twin screw. This will affect how long the material will stay in the twin screw granulator. Also, we can look at the mixing or granulation efficiency. So we can explore how the material product property or process performance is influenced by the process condition and the particle property. We can also look at more complex behavior. For example, the mixing process in continuous manufacturing. So here shows a typical continuous blending process used in continuous manufacturing line. We can use DM to simulate and explore the material, how your formulation will behave in the process. DM can also be used for process innovation. So this is a kind of a study we are currently working on. So for example, this the top one shows the existing design. By using DM, we can actually test in some new equipment or device design, how this design will perform, how this will compare with the existing one. We can look at the novel equipment design for manufacture process. So just give you a quick case study on what DM can do. Certainly for DM, there are some limitations. For example, it's quite a compute intensive. When we talk about billions of particles, it takes very long to run the simulation. So it's quite a, for some industrial process, it's not feasible at the moment. Another challenge is associated with determining the material parameter, so the input parameter used in DM. So for each simulation, you need to make sure you simulate the process for given formulations. So for that, we needed to know the material property of your formulation. And this exercise is not trivial. So it's involved detailed and comprehensive investigation to achieve that. Also, there's some challenges in modeling irregular shaped particles. Much advance has been made in the last couple of years in modeling irregular shaped particle. However, to model the real shaped particle, because each particle has different shape, is still quite a challenge. Also, there's some challenge associated with the change of a particle shape during the manufacture process. At the moment, I don't think that any model can handle this, but I know people are actually working in this area, trying to advance the DEM to consider the change of a particle shape. Now, I would like to quickly go through FEM. So FEM is a slightly different method to DEM. So in FEM, we don't treat a particle as an individual. We treat your formulation as a continuum. So it's just as a continuum material. And we use some kind of a material model to describe the mechanical behavior of this material. Then we can run the simulation or modeling on some process like a pharmaceutical tabulating process. So with the FEM, typically we needed to know the powder you are using, the formulation you are using, and the process you are using. And for a given formulation, we needed to run some kind of a model calibration experiment to obtain the material properties, material parameter that can describe can characterize the formulations. Then we put this into FEM solver. Then we can look at the material behavior during compaction, and we can also predict the property of final compact and so on. Here I gave an example. So that's a study I did when I first working with Pfizer at Cambridge University. So we use FEM, we actually managed to identify that at which place that your formulation will crack, will form this kind of failure during the pharmaceutical compaction process. So we also look at how the entrapped air. So if you have air bubble in your tablets, in the powder, when you compress it, how this will affect the property of a tablet. We can see that experimentally, we do see this kind of crack can generate during compaction. We also run FEM, so basically give a very good prediction on the potential failure place. Certainly with FEM, we can look at different tablet shape, also look at a different process condition, and we can explore the stress distribution or density distribution of the compacted tablets. Furthermore, we can also look at the temperature change during compaction process. So this is an experiment we did at the AstraZeneca using their compaction simulator with microcrystalline cellulose pH 102. So the room temperature is around 20 degrees, as you will see. Sorry. From this video, you can see once the tablet is being compressed, you ejected from the dye, there's a significant increase of the temperature. So we can using FEM to look at how the temperature change during the compaction process. So here shows a comparison of FEM and experiment data on the surface temperature. So because experimentally, we can only measure the surface temperature using thermal cameras. So generally, we can predict very well what's the temperature can generate during single compaction process. We can also look at how the process condition, like compaction speed, also tablet shape, how this will affect the temperature change. So this just shows the comparison, qualitative comparison between experiment and FEM simulation we did for flat face tablets and convex tablets. So one thing I just would like to draw your attention that from our FEM simulation, we do find that highest temperature normally is not at the surface of your tablets when you compare actually inside. So the highest temperature, the hot spots, are actually inside of the tablets. So this can be quite detrimental if you have API has a relatively low glass transition temperature or very sensitive to heat. Then it might cause some kind of a thermal instability or degradation during the manufacture process. By using FEM, we can actually explore what kind of temperature it can reach in a typical manufacture process. With FEM, we can use this for industrial scale problem, like roller compaction or die compaction. We can predict the kinetics, density, stress, strain distribution. We can also use it to indicate potential failure surface. And another strength of FEM is that it can handle complex geometries. Certainly there are some challenges as well. For example, when you run the FEM to get a convergence, computational numerical convergence, sometimes quite a challenge. Also, we need a accurate material characterization procedure to get material parameters represent the formulation we are working on. So that's just a quick discussion on the FEM. finally, I just wish to quick spend a couple of minutes on AI and machine learning. So that's quite a hot topic in modeling. So I know most of us know AI is actually advanced data-driven or information-driven method. So for AI to be effective, the first stage is learning. So we have to make sure the machine or computer can recognize and establish some kind of rule or pattern to become intelligent and can solve a task. So the machine and the computer has to learn from existing information, existing data. So therefore, the machine learning is a kind of a critical or subdomain of AI that are dedicated to develop a model to make a machine learn from existing data. So the data can be previously experienced in a digital form, or can be patterns, or can be kind of any other sort of logical informations. So there are also two types of machine learning. One is a regression, one is a classification. So for most of engineer, most of the work I'm talking about here is numerical based. So we are dealing with numerical values, the numbers. So for which the machine learning is a kind of a regression based machine learning. Also, if you're using machine learning to learn from categorical values like patterns and class, then that's what we call the classification. So you use machine learning to recognize and classify the input into different classes and groups. There are different machine learning technical algorithm has been developed and been used. For example, artificial neural network, flexible neural tree, regression tree, and so on, random forest, and so on. The large collection of different machine learning algorithm has been developed. Also, with machine learning, we can use it for feature analysis and feature selection in the pharmaceutical term. So that's what we call to identify CMAs and the CPPs, so critical material attributes or critical process parameters. We can use machine learning to do that. Certainly, in most of the machine learning technique, it involves some kind of optimization approach to find the optimal internal variables or structure or functions to achieve the health predictions. So in collaboration with several computer scientists, computer science groups, we have been exploring using bio-inspired optimization methods for pharmaceutical manufacture process. So we have looked at different optimization technique for tabulating process. So one of the interesting, we found that this kind of model can, if you give it enough data for them to train the model, and then eventually it can be used to predict the tensor strengths for a given porosity. Also, quite interestingly, it can use it to identify which parameter, input parameter, is most important, as shown here. It shows that in the tabulating process, based on the data we gave to them, the compaction pressure actually plays a significant role. The second important factor is the material. So if you change the material, you will certainly affect the tensor strength. And then it can also look at how significant the granular size. So then the size of the particle will affect the tensor strength. We also using the flexible neural tree machine learning algorithm, look at the defining process. So in this process, we provide the data with different true density and feed material particles at D50, and we roller compact the material to produce a granule with different size, and we run the dye filling at a different shoe speed. So through the optimization, the machine learning can generate a different tree structure, flexible tree structure, and it can also evaluate the efficiency, how good the prediction are, also how complex. So here the number of the numbers indicate how many nodes actually in the flexible tree structures. So use this type of model, what we can get the how the mass deposited into dye varies from different input parameters. So here shows a comparison with predicted and the existing data set. Also, we can show we can use in the machine learning model to predict the mass for given shoe speed and for given different size of the granules. As I mentioned earlier, another approach we can do with the machine learning is that to identify which feature or which input parameter is dominant in this process. For this particular example, what the machine learning tells us is that the granular size actually plays quite a significant role in the dye filling process. And then secondly is the speed. So in terms of how often this feature has been selected in building the accurate machine learning model and how good the prediction those model will be. So certain granular size and the shoe speed will be the dominant parameters. So those are some kind of interesting information we can get from machine learning. Right. Finally, just a quick wrap up. So basically what I've actually emphasized here, so for modeling the pharmaceutical product development, we have to consider different mechanical property of powder at a different stage. So then there are a wide range of material methodology, numerical model can be used, but we need to evaluate the strengths and the limitation of each method and make sure we pick up the right method for the right to simulate the process we would like to explore. Also, machine learning is a quite interesting tool as well, especially can be used to process large data set and find out the correlation rules between the input and output. Also, it can be used to identify CMAs and CPPs. There's also great potential. We can integrate different methods together because, as I mentioned earlier, the process itself is quite multi-scale. We have to we can look into combine different methods together for comprehensive model of modeling of pharmaceutical product development. I believe that those kind of computational tools will play an important part in the pharmaceutical manufacturing of the future. For example, in developing digital twins, in embracing Pharmaceutical 4.0, also some emerging manufacturing process like a continuous manufacturing and a flexible manufacturing. With that, I would like to acknowledge my colleagues have been involved in this process and the funding from pharmaceutical company and funding agencies. Thank you very much for your attention.
Okay, and thanks a lot for Charley's presentation, and it is fantastic. And so thanks, Charley. Yeah.
Right.
And so, okay, now, and I think it is my talk, and so I start the share. Okay, my, wait a moment. Okay. could you see my slides, the full screen?
Yes. Yeah.
Oh, okay, thanks. Yeah. And just now, and Dr. Zhao and Professor Charley Wu and give us the very fantastic presentation. And now I wanted to share my research. It is computational pharmaceutics, a new paradigm of drug deliveries. And first, I want to introduce a little background of drug discovery and development. And as we know, the drug discovery, we can divide it into two parts or two steps. The first step is we design the new drug molecules. In the past 50 years, we have plenty of computational tools help us to design the drug molecules, such as the bioinformatics, computer-aided drug design, or AI in drug design in recent years. And three scientists, Kaplan, Levitt, and Washer, win the Nobel Prize in 2013, Nobel Prize in Chemistry, for the development of multi-scale models for complex chemical systems. The Nobel Prize Committee also said, today the computer is just as important a tool for chemists as a test tube. And however, the second step, once we have the drug molecules, we have to prepare the from formulation or dosage form and then administrated for the patients. And however, this is the second step. Currently, we have to do the trial and error experiments in the laboratory and by the personal experience of the pharmaceutical scientist. And now we can see what is the formulation development process. We can divide this formulation development process into four steps. The first step is the pre-formulation studies. We need to know the physical chemical properties of the formulations. And then we need to select the proper excipients in the formulations, and then it is a manufacturing process. And finally, we need to do the in vitro and in vivo test. And in the recent years, we do the big data analysis to the FDA Orange books, and actually, in the Orange book, they have about 2,000 the drug molecules, and it is about 3,000 excipients and 169 different dosage forms and over 30,000 products. And this is, we can see the formulation development. It is all pharmaceuticals. It is a multidisciplinary subject, including the chemistry, material, engineering, and the clinicals. And so that's why we currently have to do try and error by personal experience of the pharmaceuticals, and a pharmaceutical scientist is time consuming and high cost. So my question it is, can we predict the pharmaceutical formulation by the computational tool? It just as the computer-aided drug design. And in the past 10 years, we try to answer these questions. And what we use main focus is solubilization strategy of the water-insoluble drugs. As we know, currently have about 40% of drug in the market, it is water-insoluble. And over 70% of drug candidates in the pipeline is water-insoluble. And it's have plenty of the solubilization strategy, such as co-solvent, salts, surfactant, cyclodextrin, particle size reduction, lipid-based systems, crystals, and amorphous solid dispersions. And actually, our lab investigate nearly all these strategies. And today, I only focus on to discuss the amorphous solid dispersion, this strategy. And solid dispersions, actually, it is dispersed the drug molecules in the hydrophilic polymers at the solid state to improve the solubility and the dissolution rate of post-soluble drugs. Currently, there have multiple different polymers can be used in the solid dispersions, and the different preparation methods, such as hot melt extrusion method or spray drying methods in the preparation. Currently, over 20 products in this technology in the market now. And here it is show it is how marketed products by these techniques. However, currently they have it is have 4 key issues to hinder the solid dispersion development. The first issue is the solid state structure of solid dispersions. As we know, since although we have 50 or 60 years in these areas, but we still don't know what is the structure, what is the interaction between the drug and the polymers. And also the second issue is aging issues during the storage period. That means it is the solid physical stability of a solid dispersion is quite poor, and the drug molecule will easily to recrystallize and in the storage period, and then the formulation fail. And the third issue, it is the dissolution mechanisms. And we know the solid dispersion can enhance the dissolution of water insoluble drug, but we don't know why. And the final question, it is in vitro in vivo correlationship, and the IVIVC also is important. So my research mainly focus on these 4 key issues. The first one, it is a solid dispersion, solid state structure of the solid dispersions. And this figure, I take it from Pharmaceutics textbook. So this is a transition theory of amorphous solid dispersions. The transition theory consider the drug molecule just like a fish trapped within the network of polymers. So, however, if we think deeply by the physical chemistry views, then we have questions. If this drug trapped within the network of polymers, and then the drug molecule were difficult to shift out and recrystallize. However, in the experiments, the drug molecule, the formulation easily to recrystallize, and then the transition, we think about the transition theory may have problems. So we use a molecular modeling method to study the solid dispersions. And as we know, the molecular modeling in the past have widely used in the computer-aided drug design. And today we use these techniques in the formulation development. And so this is this one. It is show how it is the molecular dispersion, molecular structure. And here it is we use polyethylene glycol is 10th unit, and the ibuprofen, the small crystals. And so we can see this video with the change of the temperature, the linear, the polymers will start to bend and aggregate together from random coils. The crystal structure was broken, and the drug molecule will stick at the surface of the polymer random coils. And this model gives us more reasonable explanation of the physical stability of solid dispersions. As we know, since the drug molecule just stick at the surface of the random coils, then it will easily to shift out and then aggregate and then recrystallize together. So this one can give us more reasonable explanations to the solid dispersions. And also we did the different polymers and also different ratios. We get the same results. And the second part we investigate is this physical stability of solid dispersions. As we know, the solid dispersion actually is the key issues for the solid dispersions. However, this physical stability have been influenced by many factors, such as the drug molecule factor, solubility, lipophility, melting point, and also the polymers, such as the type, the glass transition temperature, and the ratio, and also the process parameters and the storage conditions, such as temperature, relative humidities. And in the past 50 years, many scientists try to use some mathematical equations to solve these problems. However, they didn't get the good results. And so we think about this, since this is difficult to describe by the mathematical equations, we can use the machine learning to predict the physical stability of it. And as we know, we need first to think about what is the fundamental principle of the formulations. And actually in the quality based by design, it's have described what is the formulation design space. Actually, the formulation, it is a multi-dimensional combination and interaction of input variables. such as material attributes, such as the drug molecule and excipients, and also the process parameters. So since we in the past we have to do trial and error in the laboratory to look for the optimal points in the formulations. Actually, this is difficult by the trial and error in the laboratory. However, this is easily solved by the computers. As we know, in the past several years, the artificial intelligence shows great power in the Go games, as AlphaGo, and also in recent days it is AlphaFold, and it can predict the structure of the proteins. So it is fantastic progress. So today it can help us to predict the pharmaceutical formulations. So then we use the 4 steps to predict these formulations, such as the data extraction, and then is the data standardization, and then data splitting strategy, and then we build the prediction models. And here I introduce one by one. And the most difficult for to build the prediction model in the pharmaceutical formulation, it is we have no public database. So we extract the 646 data from the literature, and including the 3 months and 6 months physical stability of the solid dispersions. And also we use a molecular descriptor to describe the drug molecules and the polymers, including such as molecular weight, X log P3, and also the other parameters such as complexity log S to describe the drug and the excipients. And also the formulation, we have a small data set and with imbalanced input space due to the limited data. So we developed our own algorithms, we call MDFIS algorithms for the data splitting strategy. And also then we use a different machine learning model to build these prediction models, such as deep learning, random forest, light GBM, SVM, and anti-neural network, KNN, and decision tree, and native Bayesians. And we found that the random forest achieved the best prediction performance, is over 80% is prediction accuracy. And more importantly, this algorithm can help us to understand what is important for different factors. For example, here we know the drug loading ratio is the most significant important parameter for the physical stability of the solid dispersions. And this is very interesting, and I will explain in the next slide why it is this one. And because this one can give us the ranking of the different parameters, and then it can help us shift from the experience-based current formulation development to the theoretic science in these areas. And our results have published in the Journal of Control Release last year, and also the editor-in-chief of JCR and Professor Christine Allen also just write a comment to our progress. The title is, does artificial intelligence have the potential to transform drug formulation development? And also we did the experiments and molecular modeling to validate our prediction models. And these experiments have been it is by the Professor Ren Fuzhen and by the Huadong University of Science Technology. And we developed the 3 formulations and ratio 1:2, 1:3, and 1:5. And we got this one. It is this is the beginning of the formulation. We can see it is quite good, right? And after 3 months and 6 months, we can see in the 3 months, the 1:2 ratio formulation, it have a small peak of drug crystals. And after the 6 months, the 1:2 ratio formulations have a very clear the drug crystal peak right here. And however, 1:5, this ratio is still keep the formulation keeps stable. So it is confirms our prediction model, right? And also we did the modeling to validate, investigate why it is the different ratio. It is important. So from here we can know with the high ratio of polymers, then just now we said is the polymer forms the random coils, right? And we get the more polymers, and then we get the bigger this random coils. And then this drug molecules, the distance between the drug molecules will be far away, and then the drug molecule will difficult to aggregate together in the high polymer ratios. And then since although it is simple, however, we didn't know in the past. And the third step, we investigate the dissolution behaviors of a solid dispersion. Actually, the in vivo dissolution profile of a solid dispersion is a result of competition between dissolution and the precipitation process. And that means the dissolution profile of a solid dispersion actually will have two types. The first type is, we call it a mountain supersaturations. And the second type, we call it spring and parachute. So for the second type, we will know this drug will form the precipitate, right? And then it is the formulation fail. So we will use first we use a classification model to classify these two types of dissolution profile, and then we use regression model to build a regression model to predict this mountain supersaturation dissolution profile. And then we continue to collect the data. We including 674 dissolution profile, including 94 drugs and 9 polymers, and build the good prediction models and these feature contributions. And since we have a limited time, so we won't talk too much details. And also, this is a regression models for the mountain saturation profiles. Also, it is good. And also, we use the experiment and the modeling result to validate the prediction models. Here we use the results, experimental results from the literature. It is this drug, it is vemurafenib, and this water-insoluble drug, and including 2 formulations. One it is HPMSAS, and another one is UGG. And so we can see this HPMSAS, this formulation is much better than the UGG, right? And so we did the dissolution, did the modeling results. We can see we put this solid dispersion in the large big water bottles, and we see the UGG, the base formulation, even after 840 nanoseconds, still we have large particles in the solution. And however, for the HPMS based formulation, after 480 nanoseconds, the drug will distribute in the solutions quite uniformly. So we can see it is agree with the experiment results. And also here, this video will help us understand what is the dissolution process of the solid dispersion. This dissolution we can see. Okay, and so this is show drug molecule will quickly release from the polymer random coils, and also this random coil will start to lose, and the polymer will also dissolve in solutions. And this is the dynamic process of the dissolution process of the solid dispersions. And in the first part, we investigate in vitro in vivo correlationship. As we know, this is the most important for our formulation development. Just now, Dr. Zhao also discussed this part. And actually, also we use the PBPK models to investigate, to build the IVIVC. Since Professor Dr. Zhao already discussed this, and actually the PBPK modeling is different from traditional compartment models. They use a very complicated models, including physiological parameters, including such as lung, brain, heart, liver, kidney, and other organs. There's the physiological parameters build the mathematical models. And this model is very powerful. It can integrate the preclinical, the animal study data, and the phase 1, the healthy people, the clinical data, and also phase 2, phase 3, the patients' data together to help us understand the drug performance And in recent years, the PBPK models also to use to bridge the in vitro dissolution and in vivo performance of the pharmaceutical formulations. And so, and also our groups also use this method to investigate IVIVC of the solid dispersions. So we still use the same case and this drug to investigate the model. So here, this one, we can see this HPMSAS and the UGG, the dissolution, right? And this is the human viability. And so compared with the crystalline, the drugs, and also the best formulation, HPMAS. And we build this model, this input parameters for PBPK models. And this is the drug's physicochemical properties and also the ADME, the parameters. And this is the results of the experiment and the prediction results by PBPK models. So this dot is experimental models, and the line, it is the prediction model. So we can see the blue line, it is crystal drugs. So, and the red line, the red curve, it is the HPMAS, the formulations. So we can see this prediction model is well agreed with the experimental results. And these results have been published in the European Journal of Pharmaceutical and Biopharmaceutics recently. And our group in the past 10 years also tried different formulations such as liposomes, nanoparticles, the cyclodextrin formulations, and the gene delivery systems. Also, our group also developed the different machine learning algorithms such as user transfer learning in the prediction formulations. And also, our group developed the first global artificial intelligence platform for pharmaceutical formulations. Here, I just show you one case how to predict the solid dispersion stability. Okay, and so we just need to input the chemical structure of the drug, and also input the parameters of the process parameters. And then we submit it, and we will see the results. Okay, I just make it just go quickly, and then we will get the results of this formulation, whether it is stable or not, and the system will give you a recommendation which formulation is better. Okay, and so I will give a summary of the brief summary of our result, our research. So in the past 10 years, our group developed the integrated computational methodology for the in silico formulation design, including 3 computational tools, such as data-driven machine learning and the molecular modeling and the PBPK modeling. And also our group is published in the first book in these areas by the Wiley publisher in 2015. So we can see the computational pharmaceuticals have many benefits. It can shift the paradigm of the pharmaceuticals from experience-based subject to the theoretic science. And also it can change the try and error laboratory experiment in the laboratory to the data-driven AI. And also currently our formulation study only studies the microscopy, the properties, and then now we can investigate deeper to the molecular structure between the drug and the excipients. And of course, this area, it is a very new area. It is have many challenges in this area. The first one, it is currently we lack the user-friendly, the computational tools. So we need to develop the user-friendly good tools for our pharmaceutical formulations scientists. And also the second is the data. The data is very important for us to build the good prediction models, and how to share the data within the different pharmaceutical companies and also the laboratory is very important. And the third one, of course, the scientific talents is very important. And we, in the past 10 years, we also train the young scientists in these areas. And I introduce a little bit how it is different government authorities in this in development in these areas. Since Dr. Zhao already shared with us how it is the FDA's progress in the recent years. And actually, in last year, 2009, the FDA also initiated a big program. Is called CASA, Knowledge Aided Assessment and Structure Application. They will investigate the $50 million in these areas to start this program. And also about the European EMA and Japan, the government, they also emphasize these areas. And this year in China, the CDE also released a guidance about the model informed drug discovery and development. And actually, the industry also, it is very important. They put more interest in these areas in recent years. For example, in the UK, in past 5 years, they have a big plan. It is called, the name is Advanced Digital Design of Pharmaceutical Therapeutics. It is for 20 million British pounds in this program, and including the big pharma in the UK, such as AstraZeneca, BMS, GSK, and Pfizer, and also the top universities, Cambridge, Leeds, and Strathclyde. And also some excipient company also interested in these areas. I think that the main reason is the science, the paradigm have been shifted from the experiments to the theory, and also to the model and the simulation. And the recent years, it is data-driven, big data and machine learning and artificial intelligence. So we can see the pharmaceuticals have 2 generations in the past 60 or 70 years. So first generation is the physical pharmacy from the 1950s. And after 30 years from the 1980s, the second generation is nanomedicine and biopharmaceuticals. I think it is in the future, the computational pharmaceuticals and the computational tool will play more important roles in the pharmaceutical research. And I wanted to give 1 minute to just introduce our Computational Pharmacy Society. And the aim we want to found this society is to enhance development of computational pharmacy with the global academia, pharmaceutical industry, and the government. And this is the societies, our society, Computational Pharmacy Society, have been founded this year. And it is, if you are interested, you are welcome to join. And finally, I need to thank my collaborator and our students for the hard work and in these areas. And also I thank the funding agency, such as the Royal Society from UK, and also the FDCT, the grant agency at Macau, and also the University of Macau to support my studies. And thanks for your attention. Okay, I think now it is I finished my presentation. So could you Dr. Zhao and赵亮博士和Charley 能不能把你们的那个video 打开？对，我们看看观众听众有什么问题。对，这个地方我们收到了有一些问题。然后他们有个人通过私信给我的，也有那个现场提问的。我先就是这个问题先跟2位老师请教一下。第一个就是问题是，目前对我们计算药学的这个来说，小数据集和数据过于分散是一个很大的这个困难。然后 FDA 是有一个名叫 Open FDA 的网站，这个网站是分享了一些 FDA 汇编的一些数据。然后您能简要的给我们介绍一下这个网站吗？这个问题应该是对赵亮博士的。然后另外还有没有可能由 FDA 来引领建立一个类似于像 GitHub 这样的平台，来鼓励制药公司和研究的机构来共享一些公开的这个数据，来更好的促进这个计生药学和这个新药的这个研发，在这就是为以后我们的新药的研发来发展。 赵亮博士，您能先谈一下您的那个
对，非常感谢这个问题，问的很到位，也很 big picture。Open FDA 这个网站，我还没有特别研究过，但是我可以说一些基本的东西，就是 FDA 所有的publication， 好比外面的文章，还有我们做的 sponsor， 你看，就是刚才欧阳教授也给了一些那个也总结了一下，一共好像我看了我都没总结，你是总结了5000万美金，我觉得肯定是有这个数的，是放到这个 modeling simulation 这一块儿做研究，就是我们一方面做审评，一方面弄一些各药指南，再就是做一些研究，把这个大家普遍受到困扰的问题，就是找在世界上各个公司或者是实验室做一些研究，然后这些呢，因为我们是拿的是 taxpayers money， 纳税人的钱，所以所有的这些结果都应该是 open source。我知道做 model 这一块就是open source 还比较，就是还现在属于婴儿期，还没有到很成熟的时候。但是明年我们就会组织2个 public workshop， 单独的是做 model 的，一个2天，就是一个 PBPK 的，另外一个就是做 long-acting injectable 的，就是我同意现在的数据比较分散，就是分散的数据做药物报批的话，几乎是很难被接受。我们会把一些发表的东西，一些模型，同时呢还有一些我们现在就是 sponsor 的一些 research， 都是尽量让 open source， 就是有什么 tools， 包括 PBPK 啦，还有包括一些刚才那个吴教授讲的也有一些 open source 的那个 CFD modeling、DEM 那些 modeling 的东西，好像我们和普林斯顿有合作的，这些的 develop 这些 tool 都会 open source， 就是大家都可以用，就是我们发表的文章呢，所有的 data 什么东西都可以索取，也是都应该 open source。现在还没有特别，虽然有一个 open FDA data， 但是我不知道有多少用啊，因为内部的话，这个不是太，我还没看到太多的应用，但是是非常好的一个开始。
谢谢赵亮博士，其实这个的确是非常重要。Charley，你能不能谈一谈你的这个就是有什么建议在这一块？对。
其实刚才那个张亮博士已经回答挺好。其实我就想补充一下，就在欧洲和英国这边，就是尤其最近几年是非常提倡这个 open data 的，就是我们现在是任何英国这个基金委或欧盟Horizon 2020就是2020这个框架资助的所有项目的一些成果都是要求必须是 open access， 你的 publication 你的数据都要 open access， 尤其在英国政府是要求我们这些数据必须的 open access， 至少是10年，就是如果有人在这10年之内access 就你的数据以后就访问了你的数据以后，你这个要自动的再往后延10年，就是尽量的让这些所有的通过这个政府资助的这些项目所得到结果都是相当于这个用这种平台共享的。还有一个，我觉得刚才这提的问题还有好的，就是像现在的这个药企啊，你要让他们其实是很多数据的，
对，
很大量的数据，但是要让他们去共享或 make 他们的数据 open access， 我觉得这个难度还是非常大的。刚才欧阳教授提到的那个 UK 的adopt 的 project， 他们当时目的其中一个很重要的一个分项目就是想这些药企之前 通过材料数据的共享，
嗯
但是取得了一些进步，但是感觉通过跟他们的交流，这个难度还是比较大，当然涉及了很多原因。我就补充到这里。
对，实际上那个 Adapt 的那个他们那个项目，我也一直在跟踪，但是没有看到特别多的这个信息他们放出来，就是只能看到一些那个公开发表的这个文章啊，那些数据基本上还是看不见的，对，还是比较
现在可能会慢慢有一些，但是全部公开我估计需要很长，需要一定的时间。
对，所以对这是一个其实我们这领域刚刚开始，这也是后面其实一步一步的发展。我想这个以后就像那个计算机这个开源的这个工具，对吧 ？GitHub 上这种，其实现在已经是面向全世界，像这种，我想以后应该也会慢慢的一步一步做到这一步，对。
其实还想补充一下，就是跟这个数据共享就是相关联，就是还有你这个数据的这个标准化，其实也很重要的。你要说大伙儿都把数据共享，然后没有一个标准的话，因为你知道 AI 它是它的准确性、它的可靠性，实际靠你的数据的质量。如果你的这个数据的这个质量没有一定的标准，有时候也会造成一定的问题。这其实是一个比较
对，这个是非
常
常也是困扰 FDA 的一个问题。我是研究的那个clinical data，就是病人身上这些数据比较多。老实的讲，就是 open 有一个 FDA 有一个系统叫 FAIRS， 就是谁有什么不良反应都可以汇报。然后在那个一开始大家都很 excited， 都很激动。你看，就是说，所有的这个病人的不良反应的数据，都有分享，然后可以做各种的分析，民间也可以做。但是呢，就是对于 decision making 来讲，真的是做决策的时候，或者批不批，或者药是不是真有问题，这个很难，因为数据质量的问题，它这个数据都是事后也没有一定的实验设计，也没有一定的控制，就是事后采集的数据，就是根本就不能支持任何 regulatory decision making。好比把一个药物下架了，把这个新的什么发现了，都很难写到，第一很难写到 label 上，第二就是真的有发现有一些个副作用，根据这些 real world evidence， 我们还是需要自己掏钱做一个实验，证明这个药真的是有问题，才可以把一个药物就是下架，或者是把这个一些信息加到 label 上，否则是有很大的这个，人家肯定会告 FDA 或者告 agency， 就是药企都很有钱的，就会故意帮这个 lawyer 给你打官司，这个他们也必须要打官司的，就是在 agency 会看到很多这个 different games in play，都是因为这个就是这个数据质量的问题。我想在这个前期的数据质量，尤其是 in vitro study results，每一个实验室做出来的，甚至同一个实验室做出来不同的时间做出来的，这个可能都有一定的这个不确定性，对，都那个都不会很好的控制了。这种情况就是这个数据怎么用，就是有大数据的话，也是一个难题，很大的一个挑战，就是怎么分析出来一些规律了，可以发文章，当然可以发文章，但是真正的 make impact 就会很 difficult。
对，是，就是您说的这个，其实是刚才两位老师都讲到这个，其实我们在建这个就是 AI 的这个 model 机器学习的 model 的时候，其实也是碰到第一个最大的问题，其实就是这个数据的这个可靠性的这个问题，因为大家都知道这个目前
其实那个文献里面也是鱼龙混杂，对吧？它不都是那些数据是不是都可靠？这个真的不好说这个东西。所以我们刚开始第一步建这些 model， 提这个数据，就是要收集第一步就是很大的力气要鉴定这些数据到底是不是真的可靠，然后才能把那个数据用过来，要不然就是真的是。garbage in garbage out 的这种，就垃圾进垃圾出，你那个 useful model 就完全没有意义。这种这个其实是最关键的，对这种数据的质量和标准化的这些问题。
但是美国有一个系统有一些参考作用，好比美国一个，我不知道你们听说过一个 project 叫 Sentinel，因为美国所有的病人用药啦，还有一些不良反应情况，主要是和保险公司打交道。他就会把这个数据，每个保险公司都有大量的数据，有大概17、18个，主要的保险公司，甚至几乎把美国的人口都给覆盖了，那些病人的数据都可以拼到一块儿，做一些 post marketing analysis，就是说，如果有些病人用了某一个好比剂型的药，或者是甚至新药或者仿制药，我们就有一些药物的剂型的什么信息。如果通过这个大的数据来，刚才我讲了，就是可以从体外、体内到临床表现这么一个模型能体系建立起来的话，就可以直接想是不是哪个辅料有问题，是不是这个生产有哪个药的生产有问题，或者哪个药的剂型设计有问题，每一类或者一大类的剂型设计都有问题，这个东西都可以研究。我不知道国内是不是有这个想法，就是把所有病人的数据，还有这个一个是 claim data 就是保险公司的数据，另外一个就是 health care care record，就是就是一些医院的对于这个病人的各种反应的那些数据可以拼起来，以后确实有一个大数据的应用。就是当然这个东西不会涉及好多法律，还有个人隐私，还有这个政府的协调的那个问题，这个事儿不是太好。但是在中国可能如果政府支持的话，说不定会很好办。在国外的话，反倒不容易。
对，我是听说国内他们就是中国的这个药监局，现在他们在提倡这个药物警戒这一块，就是这一块，这现在是有法规的这个要求了。当然具体这一块我倒还不是特别的清楚。也是今年刚刚好像是那个法规出来，要求各个制药公司要做药物警戒、不良反应的这种收集啊，这种病例这一块。
是的，而且这个甚至和做剂型的人，就是在身边在深做，因为我的本科就是学的剂型，Pharmaceutics， 所以这点我了解，有好多朋友都是在这个 field 里边。就是说，当然听起来挺远，做剂型的设计和刚才吴教授讲的 CPP、CMA 是吧？那些东西都可以和这个真实世界的数据最后结合起来，就是有一个大的数据监测的模型和那个成熟的体系，就是从那个真实世界里边直接把这个 signal 给找到，警戒找到，然后再马上和剂型、和生产、和公司联系起来，直接有这么一个非常快的系统。然后同时呢，也通过学习，知道以后剂型还有生产这个 parameter 怎么来调，怎么来设计，哪个会影响这个，好比这个 stability， 刚才那个欧阳教授讲了，就是那个 solid dispersion solid amorphous dispersion 这些东西，什么会确实有一些东西，有一些药我们发现就是同一类产品，有一些就是stability 确实是有 issue， 我们就自己做一些实验，为了让产品下架，我们自己做一些实验来证明确实有这个 issue。我不好意思说哪个产品了， 就是但是你要monitor 我们最近那个和外面合作的一些实验，就大概能猜出来。
是这个实际上也是我们一直所追求的这个目标，就是希望能够从这个处方前研究到处方，然后到这个 process 这个过程，到最后的这个 in vivo 的这个过程 in vitro 到生产。如果我们都能把它建 prediction model 全部链条建起来的话，这个可能对制药业是一个非常大的这个促进的作用。这个其实这也是我们的这个一个终极目标。可是当然这个非常难了，这个这个东西
要有政府支持，我觉得在中国在国内有政府支持的话，会很快事情就会办好。
希望
包括美国的 Sentinel 几乎那么多数据，但是我们不能have direct access， 我们不能看到，只能说写一个 plan 你帮我分析一下，然后他们那边的人专门给你分析了结果，然后再给你，因为你不能自己来玩数据了，那个好多东西都不能干，因为你根本没法想象会出现什么问题，然后你给他打一次交流，几个月出去了，就是那个你想试一下问题不对，你再联系他们，几个月就出去了，很不方便。
是 OK， 好，谢谢。然后第二个问题，我是关于这个人才培养这一块的，因为我们都知道这个领域是非常新，就像Charley教授，无论是Charley教授还是赵博士，都在领域都有将近20年的这个经验了。然后那这一块其实这块的这个人才这一块，像您两位这个经验这么多，你们是觉得这个领域目前的现状和以后有什么建议没有？因为实际上也有很多的目前。包括国内有很多学生，他们特别想学这个领域，但是他们就很难找到相应的这个老师，甚至书都很少，包括文章也零星散在文章里面，很难找。所以这个Charley你先说吧，你觉得这一块这个有什么好的建议没有？给同学们。
反正就从我自己的经验来说，这个人才培养，一个是对，尤其在英国这边，尤其像我们，学生还们找老师难，我们这边是老师想找学生，也是这个挺难的。就是说，就比如说像我们有时候拿到一个项目，需要有做一些computational 方面的一些博士生或博士后也特别难，尤其是在英国这边，像找英国学生去做这种 computational 这方面，几乎难度非常大，几乎就基本上找不到有相应或者相应背景的。其实这一点其实到就是从博士培养来说，我感觉我们是从老师这个角度，这个感觉非常难找合适的这个学生。但从学生这就是你只要对这个方向感兴趣，而且像这种 motivation 这一块对我觉得是非常的，尤其对这个方向感兴趣还是还是值得进一步追求的。那么就这主要是从这个博士生培养这一块。还有我就是通过最近几年跟这个药企的接触，现在美国在这方面好像走得比较前，就是说，尤其美国有很多大的药企在过去几年都开始从高校里面招了一批，就是说毕业的博士生、类博士在做博士论文的做的就跟那个计算相关的，然后就他们到药企里面开始自己开始成立一个小的这种computational modeling 这种 group， 像这 Pfizer 这些。这些公司他们都有一些人在做这方面还挺好，但是在英国这边又觉得还相对来说比较慢一点，尤其是这个药企里面他们对这个感兴趣，但是你要是真正说要让他们自己去用这种模型，现在是就是说这种scale 的这种 staff 他们的 scientist 是基本上很少的，基本上就没有，就是说需要还是。在这个人才培养需要还是建立一个相对来说比 比较完善一点的这种系统比较好，就是像美国那种途径，在一般的大学里面博士毕业后，然后到药企去，他们到那边就开始通过这个把自己学这些 skill 啊 competing skill， 然后慢慢再带一些助手啊，这样就可以慢慢发展起来，这是一个很好的途径。
好，谢谢查理。这个实际上这一块打过广告啊，查理教授这一块我们有听讲座的这个同学，如果你们是要感兴趣，赶快跟查理教授联系。我记得前2个月您那儿还说有相关的这个博士的这个机会，对吧？
对，现在还在招呢，那个是跟那个杨森江森Pharmaceutics一块做了一个就是 Continuous Manufacturing 一个项目，就招离散元这块，现在还在招，还没招着，还一直在招，所以还是挺难的呢。
大家如果有同学感兴趣，赶快跟查理教授联系。然后如果你们要知道这个联系方式，上网查一下就 OK 了。我觉得这个你们要升博士，这一点肯定是能找到。赵博士，您分享一下您的那个。
是这样的，我看那个欧阳教授在你的 slide 里边也知道我们 division， 我现在负责这个 division 是2015年成立的，叫 Division Quantitative Method Modeling。成立了之后呢，我们大概一直在招那个 ORAS Fellow， 但是主要是以前在国内我也弄过，也有人到我们这儿做 ORAS Fellow， 至少有2、3个吧。那个，但是后来因为政策的原因，就是要进美国政府做 ORAS， 必须在过去5年之内至少有3年生活在美国，就是 Trump 执政之后新出了一条规定，所以呢，几乎把我们的人才资源从国内的人才资源给断掉了。我们只能从美国的大学招人，或者是在美国待够3年的这些人招人。但是我们已经培养了50多个，就是学生博士毕业后，然后在 FDA 在我们这个 division 做过算 postdoc 吧，但是我们的钱要比 postdoc 高好几倍，高2、3倍吧，就是那个这工资就比industry 那个 entry level 差不多，或者差稍微一点点。那个然后这50几位人呢，基本上我们都留不住，或者到了 FDA 完了之后1、2年就到，我们告诉他就是来的时候你必须得待够1年，不能说是占 FDA 一个 brand name， 或者是说我做过 modeling， 那个你就要走，但是大部分半年之内就会找到工作，因为 industry 太缺这些人了。在美国的话，那个 industry 的 pay 了，还有这个各种 package 都很好，比 FDA 要好，而且是正式工作，就是大部分人都有一半左右吧，一半稍微多一点留到了 FDA， 有一半稍微少一点去了 industry， 去了那个主要是去了新药的制药企业。现在仿制药这边呢，正在开始，我们到了这儿就是正在推这个 Computational Pharmaceutics， 还有这个 Pharmaceutics 这方面正在推，现在需要量也越来越大。但是我们那些人现在主要还是去了新药，还有这个 CRO， 还有 consulting。去新药呢，现在有2个已经做到 clinical pharmacology 的 function head， 就一些公司一些不大的公司的临床药理的主管，就是头吧，部门的头。还有一些大公司的 director， 就是 associate director。一般毕业之后，大公司为了留住这些人，因为在美国流动性很大，一般的是为了给你发高工资，都给你一个很好的 title， 就是 associate director 或者 director， 但不一定是部门的主管，主要是就是待遇好一些，为了留住人才。这方面的话，所以我想告诉如果是有学生在听众里边，或者老师教授，这方面绝对是 以后的一个方向。短板呢，就是你要是申请 funding 会有困难，因为 NIH 啦，在美国，还有我不知道国外怎么样，就是说你是单纯做 modeling 的这个 funding，在 FDA 我们这儿有一些，就是说5000万美金这么几年应该发出去了，但是那个在其他的但是这钱不多了，在美国来说不多了。你要说在 NIH 都是几亿、几十亿的发，就是所有的basic research，但是它那个里边就很少有这个 modeling， 或者做 PK、PD、Computational Pharmaceutics 这些东西，funding opportunity 很少。但是这个领域应用很大，就是横向，像我看吴教授，也最后一张 slide 也受了和好多药企有合作，因为药企确实有这个需要。这个横向的机会会很多，但是那个纵向的，从政府的这个基金来说，可能会有一些问题，这也是限制这个学生来源的一个根本原因。在美国也很少有学校有这方面的专业，像我们出道吧，都是靠个人兴趣，还有这个基本上都是从其他专业转来的。像我还有一个统计方面的 master。实际上，我做 model 从93年就开始在药科大学的时候，那时候有数学模型比赛，学校间那时候刚开始，我们还是头一批life science 那个学生，然后就这一般的出道都是从其他行当转过来的，或者从学工程的，或者学统计的，或者学这个我还没看到学计算机或者是 big data 这方面的转过来的，可能他们有更好的出路吧。现在做 machine learning， 还有 AI 非常热，每个领域都需要，就是不一定在药企了，不一定在 pharmaceutical science，但所以我还没见到从这方面转过来的人。当然，我们最近雇了3、4个 contractor， 是有这方面的这个背景，就看看最后我们能产生一些什么样的结果，有什么 make 什么样的影响，就是会随时给大家更新。
好，谢谢赵亮博士和查理老师对我们的这个分享。其实这块我也稍微聊一点我的那个就是经验，就是过去的就是10年的一点那个体会。因为我现在也是在澳门大学这边，实际上这一块也是因为在国内，我刚开始14年底15年开始在国内学术圈的时候，国内基本上很少有老师做这个领域里面，基本上我去开会，基本上一般就只有我一个报告是关于 modeling 方面的，在药剂这一块，然后就是基本上很少，然后当然大家也还是感兴趣，觉得这个东西好像很新，还是好像是不是能有点用，这个大家刚开始还是半信半疑。然后近2年我发现我毕业，就是前面也毕业了，培养了一些学生，他们有的去了别的老师那儿读博士啊，硕士读别的老师那儿读博士，包括毕业的其他的这个学生。现在我发现慢慢的国内这一块，这个学术界实际上已经有一些老师在做这块的这个工作了。然后这也是为什么今年我们就特意就是成立了这个国际计算药学协会，这个因为也是想把这个我们的这个力量整合起来。因为如果是单个人做这个，其实真的是这是一个非常庞大的这个领域，是非常难做。但是如果大家一起来做，我觉得这是一个非常是一个非常好的这个机会。而且对以后来说，其实刚才2位老师也讲，无论是从那个奖学金的这个机会、学习的这个机会，还是以后就业的机会，都是会非常好的这个东西。所以同学们如果是有感兴趣这个领域的，然后其实可以联系。然后还有一个就是说，我们其实的这个学会其实也是想里面一个很重要的就是这个信息的这个沟通。因为说实话，很多学生他们因为这真的是这个信息很多分散在这些各个角落，就是可能很难找到，包括这些教授的信息啊，这些东西他们也不太清楚。所以如果下面我们也是争取，我们现在有个群嘛，这个精神药学的这个教授的这个群，然后我们 把这些老师，如果是大家有一些相关的这些 Ph.D 的这些机会啊，包括以后就像周亮博士讲的 FDA 的有这种这个 postdoc 的这种机会啊，我觉得这个都是非常好的这种非常好的这种 training 的这个机会。然后其实分享出来，大家以后其实是对我们的young scientist 的其实是我们的博士生，包括他们以后的这个职业规划，以后这一块是个非常有帮助的。当然，最终呢，也就是促进我们新药的这个研发这一块，对这个新药的这个研发，对。
其实我觉得你成立的这个协会确实是一个很好的。很好的主意，就是刚才出来一个信息沟通以外，我觉得你们肯定也考虑在这个人才培养这一块也可以起一些作用，比如说举办一些培训班啊、交流会啊，就像这种交流会啊，这些啊，我觉得都有很好的好处。
对。
还有，我就想补充刚才那个赵博士说的这个，就是说现在人才我就说根据我个人的经验是，我原来是。刚开始介入这个就用计算方法在这个制药这块的时候，是跟辉瑞合作的。那时候我还是从工科转过来，就转过来以后，我就用 FEM 跟他们商量一下，哎，他们立马发现这个东西很感兴趣，很有用，还是有一点用处，就是很感兴趣。就是后来从那以后，就是做这个 FEA 的，就是用有些人呢，在这个药剂这个过程现在后来越来越多的人在做了。就是最近几年我开会的时候碰见很多，就是说博士生也做这个方向，然后他们就到这个药企，美国的一些药企去开始用这种方法，来帮他们做一些产品开发呀，产品的工艺和过程的优化这方面，觉得这还是就是说早期阶段可能是。很多人都是从其他专业上慢慢转向这个，其实有些东西我觉得，这个技术之间是可以就是 transferable 的。你这样不一定非要学药的再去学校学工的，就不一定要去做工科这方面，这个倒没必要。你能够在什么地方能够用到你所学的知识或你的技能，我觉得都可以做一定的，就值得尝试的。
对，这个
是的，那个我也想感谢欧阳老师把这个项目把这个概念提出来，然后还组织这么一个论坛，这个绝对是需要的，把各方面的资源和想法拼起来。另外，我觉得这个学科虽然是说起来是 Computational Pharmaceutics， 或者是甚至我以前在的那个学科主要叫 Forum-Computational。这些东西在大公司里边的处境是这样的，基本上就是会找工作很受欢迎，也是未来的方向。但是就是因为就像欧阳老师感觉到的，开会还有听受众群，还有这个我们叫 ecosystem 生态圈还没有建立起来。而有一个概念就是大家说，如果我在公司里也想在这个核心药物研发的团队里边，就首先要搞清楚一个概念，就是做 model 不是为了做 model 来做 model， 模拟数据不是为了模拟而模拟，实际上 model 是一个把目前的知识和数据和他们一样，归纳总结的，从那个数理方面归纳总结的一个科学，就是我们做 modelers 或者 data scientists not just we know the tool set， 我们呢是有这个大方向的，就是整个从这个行业的顶端朝下看的那个思维的，知道模型和数据在哪儿找，可以用到哪儿，就是到底这个新药研发或者各种药物研发，到底那些节点模型可以用到哪儿，可以从这个战略角度有一个理解，然后你就可以和别人交流，也可以在那个公司里或者政府里找到自己的位置，不能把自己归类成一个技术人员就是高级技术人员，这样的话就是不会有太多声音的。包括现在在 FDA 一开始像我弄这个 division 一开始也有很多阻力的，确实我们就是培养自己的 modeler， 就说了 就是你首先不能把自己作为一个技术人员，你是和别人在别人懂得或者是知识背景或者懂的基础上，我们还要懂另外一层，就是怎么用这个数理模型来描述现在已有的知识。这样的话，对于在团队里边对于自己的参与度和对于 decision making 做决策的时候，你的那个作用就会大很多。这就是为什么就是好比会做临床药理的，会做那个模型的他要做这个 model 很难，但是从 modeler 我们做了模型的，或者是模型科学家或者数据科学家，转到其他的转回去，他们的专门做他们的那一行非常容易。这就是为什么包括在公司里现在以后肯定是个方向，你只要懂了 model 才可以在管理层才可以就是知道这个上升的空间才可以更快更高，就是一定要塑造这么一个文化的氛围，否则我们就会被人归类成这个技术我不知道怎么做，你给我把这个数据模拟一下，给我汇报一下，这不能这样定位的。
是，这个赵亮老师说的非常对。其实我觉得，因为这是一个全新的领域，尽管刚开始有很多人还不懂，就是对这个领域。然后其实我觉得其实我就说这个应该是做 model 的这一块的这个不论是研究的这个人员，还是在像您这样在 FDA 在政府机关还是在工业界的，其实我觉得更多的要起一个引领的作用，引导他们这个做实验的人，给他们一些 guidance， 就是怎么样来指导他们来做。我觉得这一块其实是做 model 的是一个非常的强项，因为是从一个整个 big picture， 从一个大的这个方面宏观的方面可以看到，做实验很容易陷在这个细枝末节里面去，这尽管我们是一个新的这个领域，但实际上这也是为什么我觉得这个领域是非常的重要。我自己是从2011年博士毕业以后，然后就一直做这个精神药理学这个领域，然后也是就是觉得这个是一个非常以后的以后的一个非常重要的这个方向。好，林老师，您那边看看还有什么问题吗？
感谢各位教授辛苦的讲座，我暂时没有什么问题。
好，那如果这边我们没有什么问题的话，因为今天时间已经比较晚了，这个赵老师那边现在是早上9点。
我这边还好，我这边是早上。
然后
比你们这儿晚1个小时。
Charley老师这边是下午2点多。
2、3点钟。
2、3点钟。
你们是也很晚了的。
这个在国内现在是晚上10点，将近10点40、10点半过了。然后就非常感谢2位老师就是的精彩报告，然后非常感谢2位老师对我们就是给我们的这些建议这些我觉得都是非常受益匪浅。我听了这个真的是。
感谢欧阳老师组织这个会议，再次感谢。
好，谢谢大家。那我们今天就到这儿。好，也谢谢林老师的这个组织，谢谢何老师对我们的这个支持。
好的。
好，谢谢大家。
谢谢大家。
那我们就到这儿。
再见。
谢谢。拜拜。